lockdown period, we think to organize webinars on biodiversity, and henceforth we start our journey to do the job. We should know about the living and non-living ones around us. We are surrounded by different types of living beings. Some are visible and are well known to us, but small microbes are not visible through naked eyes and always mysterious and notorious. Though they are very tiny, they have immense impact on environment. Without the microbes, we are helpless. Very often, they are causing diseases, but they are economically and in terms of environment very important. Today, we are eagerly waiting to hear about marine microbial diversity and its importance. Our today's resource person is Dr. Rabindranath Bhattacharjee, former professor of life sciences, Presidency University, Kolkata, West Bengal. This is our 16th webinar on biodiversity. Each and every Saturday from 7 p.m. onward, we are going to organize a series of lectures on biodiversity. From the month of September 2020, we the members of Managing Committee of Biodiversity Series are engaged to organize the webinars on biodiversity. We are thankful to all of you for your kind response, cooperation and participation. So many participants are participating in all the sessions and gaining knowledge from the webinars. We think you will be with us for the rest of our journey. Participants who are new to our team should know about us and the motto of these webinars. Myself, Dr. Pompi Ghosh, by profession Assistant Professor of Botany. Name of my institution is Seva Bharati Mahavidyalaya. It is situated in a small village near Jhargram city of West Bengal. There are participants from school level to research level and faculty members, and our resource persons are doing a very good job by disseminating the information and knowledge to the participants. We all are doing this work voluntarily. Dr. Devobrata Das, admin, Flora and Fauna Group, is really a very energetic and enthusiastic person. By profession, he is Associate Professor of Botany. Name of his institute is Government General Degree College Lalgore, shortly GGDC Lalgore. This is newly established government college under Government of West Bengal. This college is also situated beside a village, Lalgore, under Jhargram district of West Bengal. Dr. Das is a very good researcher and spent so many years in research at Central National Herbarium, Botanical Survey of India, Howrah, as junior and senior research fellow before joining this job. Our another partner is Sri Amolesh Jana, Secretary, Drishtikon Organization. He is a retired person but likes to manage diverse elements of biodiversity for the purpose of earning and making job opportunities for community development. Lastly, our smallest member is Attoja Abhirupa Das. She is quite enthusiastic to share her knowledge about environment and biodiversity with others. Today, we have one announcement. I am very happy to announce you that we are going to organize a national level conference on biodiversity. It is our 3-day program, 26th March to 28th March 2021. After the lecture of the resource person, we will elaborately discuss about the conference. Now, I welcome Sri Raj Shankar Das, one of our participant and member of Flora and Fauna Asia Facebook group, to deliver the welcome address of our today's resource person. Sir Raj Shankar Das, can you hear me? Sir Raj Shankar Das.
Yeah, I can hear you. Thank you very much, ma'am. Thank you, Dr. Pampi Ghosh, ma'am, Assistant Professor of Shiva Bharati. Can you hear me?
Sir, more loudly. Sir,
Okay.
more loudly, please.
Thank you very much, ma'am. Thank you, Dr. Pampi Ghosh, Assistant Professor of Shiva Bharati Mahavidyalay, Kapgadi Jhargram, for your nice introduction on Biodiversity 16. Myself, Mr. Raj Shankar Das, member Flora and Fauna Asia Facebook group. On behalf of Dr. Devobrata Das, admin Flora and Fauna Asia Facebook group, I welcome all the participants and praise resource person for the Biodiversity Webinar 16. Today's topic is marine microbial diversity and its importance. And the resource person is Dr. Rabindranath Bhattacharjee. Sir is former professor of life science, Presidency University, Kolkata. Sir did his PhD from the University of Burdwan, West Bengal, in the year 1988. His teaching experience, 24 years. Research experience, 36 years. His field of research, legume rhizome symbiosis, green synthesis of silver nanoparticles and its bioefficacy. He is life member of Indian Science Congress Association, Association of Microbiologists of India, Botanical Society of Bengal, Plant Physiology Forum. He is also reviewer of many reputed journals, including Elsewhere, Springer, and Taylor and Francis. Now, he is serving as guest professor in several institutions. The University of Burdwan, Hooghly Mohsin College, Vivekananda Institute of Biotechnology, Nimpit. His publications, 39 research papers in many internationally reputed journals, 16 book chapters. He is also elected fellow of Linnean Society of London. What I am saying, sir, is more than that. I have great privilege to present myself in this auspicious evening as a member of Flora and Fauna Asia Facebook group and participant of this webinar. Sir, please proceed. We are eagerly waiting to hear you.
Can you see me?
Yes, sir.
Okay.
Yes, sir. It is visible.
Yes, sir, we can see you.
Good evening. Welcome to all of you. I'm first acknowledging Professor Raj Shankar Das Rajsankar Das, Flora and Fauna member, who introduced me among you. First of all, I also thank to the members of Flora and Fauna Asia Facebook group. I am grateful to Dr. Devabruta Das, Head, Department of Botany, Lalgarh Government College, for giving me a chance to share my topic before the learned persons. I am also thankful to Amolesh Jana, Secretary, Dristikon, Dr. B. Choudhury, Principal, Vice Principal, Seva Bharati Mahavidyalaya, Jhargram, and Dr. Mrs. Pompi Ghosh, Head Department of Botany, Seva Bharati Mahavidyalaya, Kapgadi. The topic which I am going to share with you is marine microbial diversity and its importance. Well, we are living in a beautiful planet, the Earth, which is more popularly known as the blue planet. If you look this slide, it is found that we are residing in an island and surrounded by the blue water. That's why it is called the blue planet. I'm saying that this is a beautiful planet, but we are facing presently the unprecedented events in the world, and we are in the stumbling block conditions due to the coronavirus diseases. I think, which is the only single term all the person know these terms. In every corner of the world, this term is come to all the persons. These slides give the information of class 4 standard. All we know that our earth surface is covered by land, 1/4 is covered, and rest, the 3/4 is covered by the water. Now, if we consider that how much water distribution on the earth in respect of this 3/4 water, it is found that 97% oceans, 97% water is covered by the oceans, and rest 3% by the glaciers, polar ice caps, fresh waters, that means the rivers, ponds, such and such. Now we come to the microbes. Microbes, all you know that it falls within the diameter, within the dimension below the 0.1 millimeter. That is below the 0.1 millimeter dimensions is called the microbes, because we have to see the microbes with the microscopes. And it was found that the microbes, we know that the earth was formed around 4.5 billion years ago. But as at that time the atmosphere is so hostile, within 1 billion years, there's no life was formed. The first life appeared in between 3.8 to 3.5 billion years ago. Now the question comes, what type of organisms was the first microbes? It is definitely doubtful, but most of the scientists opinion that the first life was most probably the heterotrophic microorganisms, and it is believed that most of the life forms evolved initially in the marine habitats. In the figure, what is found in this slide is that it is the thought of the Carl Woese presentations. Carl Woese, great taxonomist of bacterial taxonomist, he, on the basis of the RNA sequencing, he viewed that the bacteria are not closer to the eukaryota, which was thought earlier. Nowadays, it is thought that archaea is more closer to eukarya, not the bacteria. This is the present thought of microbes and all the domains. Oh, this slide is, the picture is of the Anthony van Leeuwenhoek of the Netherlands. He was the first persons who discovered the microbes from his own teeth and the water by his own microscopes. But it should not be confused that he constructed the microscope or he invented the microscope first. Microscope was invented earlier. Anton van Leeuwenhoek, he discovered the microbes in his own microscopes, and that's why he is more popularly known as the discoverer of the microbial world. The term microbes was not at that time. That's why he named his studies the animalia. What he have seen in the microscope, he told it is the animalia, because at that time, that means at 1676, the term microbe was not there in the microbial dictionary or scientific dictionary. The first microbes in the dictionary came in 1878 by the one French military physician and surgeon, the Charles C. Dillot, who first coined the term microbes. Now we will give some of the basic information regarding the oceans. Ocean is vast. The average depth of the ocean is 4,000 meters. That means about the 4 kilometers. That is 4 kilometers. The maximum depth of the ocean is 11,000 meters. That is 11 kilometers. And salinity average 3.5 parts per thousand, and average pH 8.3 to 8.5, and temperature below 100 meters, it is 0 to 5 degrees centigrade. Now the different zones of the sea. The sea can be divided into two zones. One is the littoral zone and second one is the open sea zone. The littoral zone is the part of the sea which is closer to the shore. That means the seashore is more popularly known as the littoral zone. And in this zone, the total coverage of the sea water is about 7% of the total sea. And most of the marine life found in the coastal habitats. That is, the coastal habitat is the marine life, source of the marine life. Now the open sea. Open sea area outside the coastal area is called the open sea, but it never includes the sea floor. The sea floor never includes the open sea. So the total sea can be divided into the two zones. One is the littoral zone, which is the seashore, and one is the open sea zone, and below the open sea zone is the sea floor. Now we'll go to the different regions of the open sea. We will not count the littoral zone, but we will go to the different zones of the open seas. The first from the upper surface, it is the epipelagic zone. That means the upper open oceans. From the upper surface down to the about 2,000 meters, this zone is called the epipelagic zone. The upper 200 meter zone is called the epipelagic zone. This zone is characterized by that the most light can be entered up to this zone. That means up to 2000 meter, up to 200 meter, the sunlight can be entered, and that's why the enough photosynthetic organism can live in these zones. Below this zone, that means below this epipelagic zones, there is the mesopelagic zones, which is covered from 200 meters down to 1000 meter, and this light, very dim light enters, very diffuse light entered in this area, so this is not sufficient for photosynthetic capacity. Below the mesopelagic zone is the bathypelagic zone, and these zones is from 1000 meter down to 4000 meter, and this is also called the midnight zone. That means it's a very black zones, and only some bioluminescent organism may present here. No other living plants, no living plants can be reside here. So this is the third zone, and below the bathypelagic zone is the absopelagic zones. It is about 4000 meter to 6000 meter. It is also dark zones, and the last zone is the hadopelagic zones. It is greater than 6000 meter in depth, and it is about to 11,000 meter depth. And this zone is 90% is unknown. That means the scientific study is not done properly. There are a lot of constraints that they are to study these zones. Very few species may present, and this species is present mainly in the hydrothermal vents. We will come to the hydrothermal vents later on, but very few species which are present generally in the hydrothermal vents. Now the deep sea. I mentioned that deep sea is greater than 1000 meter length, and from 1000 meter to 6000 meter, it is the deep sea. We can say that deep sea. It is relatively biologically inactive. It is relatively biologically inactive because the sunlight cannot penetrate in this area, and it can also be found that the 70% of all ocean water is in the deep sea. Organisms in this area, why organisms in this area are very low? Because the organisms have to face a lot of environmental constraints. That is, it is the low temperature. We know that in this region the ocean temperature is 2 degrees centigrade, about 2 degrees centigrade, and it is high pressure, high hydrostatic pressure, and the low nutrient level, that is oligonutrient levels. So that's why the deep sea are the constraints for the living organisms. Now regarding the chemical elements present in the oceans. The oceans contains almost every naturally occurring chemicals, but the ratio is the variable. It is found that the 55% of the minerals are from the chloride, and then sodium, then magnesium. Rest are very negligible amount. So apart from hydrogen and oxygen, maximally, rather more than 50% is the chlorine. Now we come to the hydrothermal vents. Hydrothermal vents, it's a looks like a fissure on the seafloor. It looks like a fissure in the seafloor, which geothermally heated water discharge. It is generally found in the volcanic active places, areas where the tectonic plates are moving apart at the spreading centers, ocean basins, and the hot spots. And this in this hydrothermal vents. These hydrothermal vents can be categorized into two. Number one is that warm vents. That means the warm vents means which emit the hydrothermal fluid at the temperature of 6 to 23 degrees centigrades. And that means these vents are warmer, and some organism can be resided here. And the other vents, other hydrothermal vents are more popularly known as the hot vents or black smokers, because yes, first one is the warm vents, which emit the hydrothermal fluids at the temperature 6 to 23 degrees centigrade into the sea, that is where temperature is at 2 to 3 degrees centigrade. So in this vent, microorganisms can be decided. And the other one, the hot vents, it is called also the black smokers, because mineral-rich hot water form a dark cloud. So the picture is the dark cloud is there, and the white, it is also the warm vents can also be called the warm white vents. So this black spot, the black spot cannot be within this area, no lives can be resided. What types of microbes can be present in this, the hydrothermal vents, particularly the warm vents? It is the sulfur oxidizing chemolithotropic bacteria. We know what is chemolithotropic bacteria. So sulfur oxidizing chemolithotropic bacteria can be present here, and that is the thiobacillus, thiopteris, bacteria, such and such organisms which are present in the hydrothermal vents, in the particularly in the warm vents or white vents. Now, very common things about the microbes, the marine microbes and their general characteristics, features, size. It is found that in contrast to the soil or the water, that means the fresh water, the marine microbes are generally in the smaller in size, particularly the 0.5 micrometer to 1 micrometer in their longest diameters. In generally, they are the smaller in contrast to the soil and the or fresh water. There are some exceptions in the world. In this case, the two bacteria which is more popularly the largest bacteria in the world, that can be found in the marine water. One is the Eupolopicium phycelsoni. As the name suggests, phycelsoni. So it lives in the intestine of the surgeon fish. It lives in the intestines of the surgeon fish. That's why the name is Eupolopicium phycelsoni, and it can be seen in the naked eye. It can be seen in the naked eye. To some extent, it can be seen in the naked eye. And the last or the second one is the Thiomargarcia namibiensis. And as the name suggests, the namibiensis, so it is found in the coastal belt of the Namibia. So these are the largest bacteria in the world, which is found in the marine and which is not found in the terrestrial condition and the other conditions also. So largest microbes and the smallest microbes both can be found in the marine conditions. And some basic things about the microbes, what I am telling, that is the gram nature of the microbes. Gram nature is very important. It is astonishingly that the most of the bacteria found in the sea water, it is the gram negative in nature. Why it is gram negative? I come to the later on. But the thing is that their wall is better adapted for the survival in the marine environment. For the marine environment, for their survival, the gram-negative cell wall is more important than the gram-positive bacteria. And different types of examples are there: Pseudomonas oceani, Flavobacterium. These are the gram-negative bacteria, and all you know that these gram-negative bacteria can also found in the soil conditions, in other conditions also. So that's why that they are not the obligatory to the marine conditions. They are not obligatory to the marine conditions. They are facultatively marine conditions. They can faculty marine conditions. That means they can live both in the soil as well as in the marine water. The species is the same, and the strains may be the difference. As because you know the Pseudomonas Flavobacterium is very common. The gram-positive bacteria found normally in the marine sediments, a very few number of the gram-positive bacteria. And for example, it's the Bacillus subtilis also. Bacillus subtilis is very common bacteria. Bacillus subtilis is very common bacteria, but the strain of this Bacillus subtilis is the GLP20b, and which can be found in the Indian Oceans. And the other gram-negative bacteria, Vibrio mimix, it is also found in the. Now the temperature and the pressure relationships among the bacteria. Temperature and the pressure relationship among the bacteria. It is important that the mostly they are psychrophilic or the psychrotrophic. Psychrophilic, you know that psychrophilic means which require the low temperatures, otherwise they cannot survive. And psychrotrophic, that means they are basically a not the cool temperature loving, but they can survive in the cold temperature. That's why they are called the psychrotrophic or psychrotolerant, whatever may be. That this is both the same term. So marine bacteria mostly psychrophilic or psychrotrophic, as about 90% marine environment is the below 5 degrees centigrade. So that's why they must have to be psychrophilic or psychrotropic. And the hydrostatic spacer, that means the barophilic conditions. Barophilic conditions, you know that the 10 millimeter depth, 10 meter depth. In every 10 meter depth, the one atmospheric pressure or one hydrostatic pressure is occurred. So total, I have mentioned the total depth about 11,000 meter. So total hydrostatic pressure might be 1100 hydrostatic pressures. So because in every 10 meter depth, one hydrostatic pressure is increased. So hydrostatic pressure increase in 1 unit per 10 meter. So most marine bacteria are either barophilic or barotolerant. That means same thing. Barophilic means it requires the high pressure, or barotolerant means is not the, basically it's not barophilic, but they can survive in the barophilic, in the high pressure conditions. Example of some barophilic bacteria is the Photobacterium, Salmonella benthica, and definitely both are the gram-negative bacteria. Some Archaea, it's a very primitive one. Archaea also present in the barophilic conditions, in the barotolerant or the barophilic conditions. And extreme barophiles, this is the world's largest extreme barophile bacteria, and that is Cloella. Cloella hadriensis can overcome 1035 atmospheric pressures. So it's a definitely gram-negative bacteria. So this is the world most barotolerant gram-negative bacteria. Now these are some photographs of the bacteria. Now it's very common of the as microbes. What are the aerobic and anaerobic? You know what is aerobic and what is anaerobic, and what are the microorganisms present in the sea. That is generally they are aerobic. Generally they are aerobic bacteria. Some also the facultative anaerobic. Some are facultative anaerobic, and you also know that what is facultative anaerobic. Facultative anaerobic means that they are basically aerobic, but they can also survive in the condition of the anaerobic conditions. So these are the aerobic bacteria, particularly the alkaligens. Alkaligens, different species of alkaligens, Pseudomonas marina, Pseudomonas nautica. These are the aerobic bacteria. Might be facultative anaerobic bacteria, that means the Ultramonas, Salmonella. Few are the obligate anaerobic. Few are the obligate anaerobic means that they need the anaerobic conditions. These are the Cyclobacter alternatus. These are the obligate anaerobic bacteria. Now we come to the actinomycetes for the marine environment. The actinomycetes, we know that actinomycetes is a very important group among the bacteria, because this is also called the filamentous bacteria, because most actinomycetes, it looks like a fungal hyphae. It's look a fungal mycelium, though it's very thin mycelium like structures. It's important because actinomycetes give the most of the biologically active potent compounds. In any case, it may be the marine conditions, it may be in the terrestrial conditions. Actinomycetes, it's a very industrially potent microorganisms. So actinomycetes from the marine environment are not studied earlier, so they are the recent studies. Actinomycetes are the major source of the many secondary metabolites, including the antibiotics. And we know that the antibiotics which are used today, about the 4,000 antibiotics are present, which are presently in the market. 4,000 antibiotics present in the market, and out of which only 50 are using by the doctors most frequently. And the Streptomyces genus, the single this genus produce the more than 50% antibiotics, Actinomyces isolated from the marine environment also show the different beneficial activities. I will come to the later on. Streptomyces chiabensis from the marine sediment of the Bay of Bengal show the cytotoxic activity. What are the different compounds or different properties present in the marine activities? The first is the anti-cancer activity. The chinooxin A, chinooxins A and B isolated from the marine Streptomyces. The strain number M045 showed anti-cancer activity. Antifungal activity, it's Karthik et al. 2010 isolated Streptomyces from Nicobar marine sediment showed the potent antifungal activity and cytotoxic activity. It is Amycoptopsis alba. Alba means you know very well, it looks like white. The strain number, the DVRT4 showed the antimicrobial and cytotoxic activity, and antimicrobial activity also given by other strains of the other bacteria, other Streptomyces parvulus. It was discovered by Saik et al. 2017 from the Bay of Bengal. So the potent antimicrobial activity. Now the marine fungi also studied give the little attentions, and it is also can be said that the most of the marine species are isolated from the sea source, and few are from the deep sea, mostly from the sea source area. It is also can be said that the biodiversity of the fungal life drastically decreases with the increase of the salt concentrations. I have mentioned the salt concentration in generally it is 3.5 ppm pp parts per thousand. So as the increase of the salt, the frequency of the fungus will be low. The total fungal so-called isolated is 444, and it is also amazingly that the most of the fungus is from the actinomycetes. Most of the fungus it is actinomycetes, and rest is the basidomycete and the zygomycotina and the mytosporic spongi, that means the Dendromycetes fungus. Some are deep sea fungi are also there. I have mentioned that most of the fungi are from the seashore area, that means the littoral zone area. In the deep sea area, 5 species of indigenous deep sea filamentous fungi are present, and these are the names of the deep sea. But it is also found that some yeast genera are present in the deep sea, and it is Saccharomyces, Candidata, Torolopsis, Cryptococcus, and these are found in the deep sea regions. Now, what are the importance of the marine fungi? What are the importance of the marine fungi? Fungi, they are the major the decomposer of the woody and herbaceous substrate in the marine ecosystems, and they release the minerals from ingredients from these decayed organisms, and they are also degradation of the dead animal and the animal parts. Very few are the pathogenics. Few are pathogenics. Some are symbiotic with the other organisms, and they are important. Hello.
PPT is not. PPT.
Hello.
PPT is not presenting. PPT is not
presenting. There is what? PPT আসছে না বলছে, PPT আসছে না বলছে। আসছে? আসেনি?
Go to the presentation mode. Go to the presentation mode. Yes sir, go to the PPT. Okay sir.
হ্যাঁ, দেখা যাচ্ছে?
Yes.
হ্যাঁ, দেখা যাচ্ছে?
Okay, now okay, but go to the present slide. Scroll.
এবার দেখা যাচ্ছে?
হ্যাঁ, আছে। This is the first slide, sir. Go to the
This is the first slide.
This is the first slide. So go to the presentation slide.
Very good.
Sir, go to marine fungi slide.
হ্যাঁ, ঠিক আছে, আমি
Okay sir.
নাম লিখে নিচ্ছি। This is marine fungi slide.
Yes.
Hello,
Yes, start now.
শোনা যাচ্ছে?
Yes. Start here, please. Okay, sir. Okay. Proceed.
Hmm. We are in the importance of the marine fungi. We have mentioned that they are the major decomposers of the woody and herbaceous substrate in the marine ecosystems. And what I am telling that they can decolorize the on dye, poly R 478 and the azure dye. So dye can be decolorized. It's an important property of the marine fungi. And this is also, they have some property of the antiviral property. Antiviral property, particularly that their replication property can be inhibited by the marine fungi, and this is effective against the H1N1 H1N1 virus, herpes virus, and respiratory syndrome by common fungi, Aspergillus and Clostridium. I am also again mentioning that Aspergillus, it's a very common fungi. Bacteria, it's a Clostridium, it is written wrong. Aspergillus, it's a common fungi, but they have the different strains of Aspergillus. Now the marine virus, it is to be noted that out of the total microbes, and you know that the virus definitely under the microbes, and out of the total microbes, regarding the biomass of the microbes is definitely the bacteria. Biomass of the marine organisms, definitely the maximum biomass occurred by the virus, definitely. And but in respect of the number, the virus is the highest. In the side picture, it is clear that the blue one is second blue one is the number of the viruses, and the first yellow one is the number of the bacteria in respect of the biomass. And in case of the second one, the blue color, it is the number of individuals in respect of the virus. So huge number of virus is present in the marine environment. And if we take a teaspoon of the seawater, it contains 10 million marine virus. And it is also to be noted that most of the virus are the bacteriophages. What is bacteriophages? All you know that. And they destroy the marine bacteria, and control the growth of the phytoplankton at the base of the marine food web. That means that they have some link with the marine food web. How this virus can be studied? How this virus hunting can be studied? It is by transmission electron microscope. It is studied, but this method is nowadays not so done, because it is very tedious, it is expensive, it is time consuming, it's lack precision, and it needs some technical sophistications. That's why the TM analysis for the viral counting is not done here. Presently, some strain, the name of the strain is the DP1.
Victoria. প্রথমে টিপে দিতে হবে।
Participants, please wait. Sir is disconnected.
It's okay, ma'am. Thank you.
হ্যাঁ আপনি বেরিয়ে গেছেন।
Wait a bit. He will join very soon.
হ্যালো শুনতে পাচ্ছেন?
It's a very illuminating saying, ma'am.
Due to poor network connection, sir is disconnected. So now sir is coming. Wait.
Not audible, sir. Anything is not audible.
Hello. Hello. Now it is audible.
Yes, sir.
Yes, yes, now audible.
Now the hello.
Sir
Yes,
is,
yes,
sir is
yes.
presenting. Sir
Hello.
is presenting.
Yes, yes, sir. Yes, sir.
শোনা
proceed.
যাচ্ছে?
Yes, yes.
Is it audible?
Okay, okay. Okay, sir,
audible,
proceed.
sir.
Yes, audible and clearly visible, sir.
What is Tata?
Just screen, sir. It's okay.
Start, sir, sir.
Hello.
হ্যাঁ ঠিক আছে স্যার শুরু করুন
Acha. Earlier I had mentioned that the TM analysis was the only method for counting the virus, but due to some technical precisions and cost effective, the TM analysis was not done for the counting of the viruses, particularly the marine viruses. Nowadays, the strains, the DAPI and the SYBR green strains, they are used for the enumeration of the viruses through the efflorescence microscopy. The picture in the right hand side, it is the there is some large dots and there is very small dots. Through the efflorescence microscopy and with the help of this dye, it is found that the large dots are the bacteria and the very small dots are the virus. And virus mainly infect the bacteria, archaea, cyanobacteria, and also it is found that the tail phages means that those bacteria, those viruses which have the tail portion, those type of virus are present in the marine virus, mainly present in the marine virus. Riju. % biomass of the each day in the oceans, and by this way the harmful algal blooms can be killed by these viruses. So viruses are responsible for the rapid destruction of the harmful algal blooms which kill the other marine lives. Now the importance of the virus on the horizontal gene transfer, it's an important one. We know the horizontal gene transfers. It is done by the viruses. It can be done by the viruses. Phages, different types of phages are the important vectors for the horizontal gene transfers in the marine ecosystems. Cyanophage, particularly the two bacteria, two cyanobacteria, the Staphylococcus and the Pleurococcus, they carry the structural genes for the photosynthetic reaction centers with the help of this horizontal gene transfers. That means the cyanophage, they take the gene, some portions of the genes from the Staphylococcus to the Pleurococcus, or vice versa, Pleurococcus to Staphylococcus to the photosynthetic reaction centers. So this is the horizontal gene transfers, and this way the evolution can be done. This way we know that the horizontal transfer, through horizontal transfer, evolution can be done. So through in the marine systems also, by the horizontal gene transfer, by the virus, the evolutions can be done. Now the marine archaea, we know that archaea means it's a primitive one. Different types of archaea, have a definite role in the biogeochemical cycle also, and the main role is the methanogenic archaea, that means which are able to form the methane. Many of which bacteria, as they are a major source of methane, such environments can play a pivotal role. So what I want to say that, why I mean to say that the archaea are the major source of the methane group. And among the different classes of the archaea, one group is there, the Cranoarchaeota. These groups are mainly responsible for the formation of the methane in the ocean. Now the marine algae. Marine algae, it's a algae, that means we know in case of the macroalgae, we know that the group Phaeophyceae and the group the Rhodophyceae, most of the marine algae, and they have the potent source of the different bioactive molecules. And it is also known that any stress condition, in any stress conditions, there should be some bioactive molecules, because they have to overcome these adverse situations. So that's why the marine algae, that's why in marine organism, they are able to produce some definitely the bioactive molecules. That's why the marine algae, from the marine algae we can get the agar-agar and other things. Whatever may be in respect of the cyanobacteria in the marine conditions, it is the first life which produce the oxygens and keep the earth aerobic conditions. So aerobic conditions appeared due to the activity of the cyanobacteria, and which most probably arise in the marine conditions. Now, what are the different cyanobacteria, which is very important? Number one is it's a Prochlorococcus, the major contributor of the atmospheric oxygens. This is the single cell organism. This is the single cell organism, cyanobacterium, and this is the major contributors of the atmospheric oxygens. And you also know that the marine diatoms are there. Different diatoms, it's a very interesting one. The diatoms are there. I am not mentioning the details name of the diatoms. So plentiful of diatoms you can find in the marine, particularly in the surface region. Particularly in the surface region, below the not the absolutely surface region, below the 1 meter region, it's the marine diatoms are present. Now the nitrogen fixing cyanobacteria, and all we know that nitrogen fixing bacteria, the common name is definitely the Rhizobium azotobacter is the common name. We know very well. Biological nitrogen fixation of nitrogen, ultra oligotropic waters, that means in the sea water. Ultra oligotropic waters where the nutrient conditions is very poor. That was discovered in Jeep et al. 2008. Earlier, it was thought that the Cyanobacteria richelieu is the only microbes which can fix a large amount of the nitrogen. Nitrogen. They resides as an endosymbiont with the others others diatoms, and they have the NIP gene similarities 98% with the NIP H sequence of the common nitrogen fixing organism, that is the azospirillum. That is the common azospirillum is the model of the nitrogen fixing organisms which have the 98.8% similarities of the NIP genes of this Richelieu intracellular entities. Now, this they fix the nitrogen both spatially and time distributions. It is a uncommon term. It's both spatially and time distribution. All we know that the nitrogen fixing enzyme, that means the nitrogen, it's an anaerobic enzyme. That means with the oxygen with the air, this nitrogen enzyme is inactivated. This enzyme is inactivated. But most of the organisms which can fix the nitrogen, that means the Rhizobacter, Azotobacter, both are aerobic organisms. The Cyanobacter richelieu, what I am mentioning here, the richelieu, it's also aerobic one. That means the enzyme is the anaerobic, but the bacteria or the cyanobacteria is the aerobic. So it's a very difficult conditions. If they will take the oxygen, nitrogenous enzyme will be died, and if they do not take the oxygens, they will die. That means it is very difficult conditions. That's why they can fix the nitrogen both spatial and time distribution. Means spatial distribution means that they have the heterocyst. They have the heterocyst which within heterocyst there is anaerobic conditions is there. Anaerobic condition is there where where the photosystem 2 is also absent, because it is the oxygen evolving area, photosystem 2 of the photosynthesis. So due to these conditions, heterocyst can fix the atmospheric nitrogen, and they can maintain the anaerobic, not the anaerobic condition, but those anaerobic conditions by which the bacteria can fix or cyanobacteria can fix the atmospheric nitrogen. And the temporal, second one is the temporal distribution. Temporal distribution means that the time distributions. That means the photosynthesis by the cyanobacteria are done particularly in the day, particularly in the daytime. After the afternoon, the operant system of the photosynthetic one is switched off, and the nitrogenous enzyme system operant, they are on. So up to this way, by this way they manage their anaerobic and aerobic conditions. That means after afternoon they switch on the nitrogenous operant, and they can fix the atmospheric nitrogen. And again in the dawn they switch off the nitrogenous operant and switch on the photosynthetic operation. That means that by maintaining the time, they can synthesize, they can fix the atmospheric nitrogen. And another one is I mentioned the spatial one. That means that they can synthesize, they can fix the atmospheric nitrogen in the heterocyst one. So Richelieu intracellular entities, they can fix the atmospheric nitrogen in both conditions by spatial and is the time distributions. The marine protozoa is a lot of marine protozoa is present. They can tolerate up to 10% salt concentrations. They mostly are the flagellate rhizopods and ciliates. The flagellate Coxylitheophoridae are the smallest planktonic protozoa in the sea and achieve the constituents of the marine plankton. So this is about the protozoa, and protozoa helps in the grazing of the bacteria, and by grazing of the bacteria they increase the mineralization and making the nutrients available for the other organisms. They also absorb the carbon dioxide in the water body and making the less harmful to the other organisms. Now the approaches to the marine bacteria, study of the approaches of the marine bacteria. One thing is that you have to isolate the bacteria in pure culture. It is an essential step in every study of the microbes. So isolation in the pure culture, it is very important in respect of the marine bacteria also. Earlier it is found that the more colony-forming units, CFU, provide inadequate descriptions because traditional cultivation method do not mimic the real environment. So traditional cultivation process is not applicable for the marine bacteria. Marine bacteria are cultured in filtered autoclave seawater following the dilution extreme. That means you have to make a lot of dilutions. Then you can culture, and several different types of media are tested and few are applicable, and other one is the enrichment technique. So from this slide it can be said that the isolations is a very important step. The earlier more colony forming unit was studied, but it was not applicable in case of the marine bacteria, and the main it is the different media, different enrichment culture technique are useful for the marine stratosphere. Adaptation of the biotolerant marine bacteria. I have mentioned in case of the biotolerant why some bacteria can tolerate the high pressures. Because the three regions or more than three regions are there. One region is the enzyme folding. They fold the enzyme in such a way that the hydrostatic pressures cannot affect their mood. So folding of the enzymes is very important one. Second one is the fatty acids in the membrane. Fatty acids in the membranes. I mentioned in the here that most of the gram-negative bacteria, that means the 90% of the gram-negative bacteria. Why the gram-negative bacteria? As because they are in respect of the marine conditions, the organism grown in the high increased pressure, they have high unsaturated fatty acids in the cytoplasmic membrane. In the membrane, they have the more unsaturated fatty acids, and which help the membrane less likely to gel, and the function of the membrane will be better for these types of unsaturated presence of due to the presence of unsaturated fatty acids, and they also shorten the length of the fatty acid by which the membrane function is will be better. And another important thing is that the outer membrane protein OMPH protein, it's a one type of the porine protein. It's a one type. We know the porine protein. Porine channel is there, and this normal porine protein is helpful. Normal porine protein means this protein is helpful for the normal common bacteria. But in the membrane, there is a special type of porine protein which is called the OMPH protein, and this protein is helpful proper functioning of the membrane, and this protein helps for the diffusion of the organic molecules. And how these halophilic organisms survive in this high salt concentration? There are two strategies there. One is called the biocompatible solute strategy, and second one is called the salt in strategy. The first strategy is called the biocompatible solute strategy. That is, the majority of the bacteria and the other organisms, they synthesize some chemicals, particularly some special type of osmoprotectants, that is polyols, betaines, and ectoines, which are synthesized from the environmental conditions, and due to the synthesis of this type of the osmoprotectants, they can overcome the osmotic stress in the high salt concentration. And the second one is the salting strategy. This is a very common practice. Salting strategy means they incorporate into their cells. Hello.
Okay, continue, sir. Continue. No problem. Continue, sir.
Incorporate their cells by potassium salt and release their sodium salt from their cells. So from this is called the salting strategy. It is found in extreme halophilic archaea, Halobacterium saliniarum, and the Eubacterium, the Salinobacter rubra. The importance of the marine organisms. The first one is the enzyme. The seawater, which is chemically closer to human blood plasma. Seawater, which is chemically closer to blood plasma, could provide microbial nutrients, in particular enzymes. Those enzymes are safe, having no or low, less side effects or less toxicity, which is used for therapeutic application because they are closer to human blood plasma. And decomposers, they play a crucial role in the decomposition of the organic matter, and it's a nutrient cyclic. And food, it is also important. They particularly they serve as the food for the bottom living organisms. Now, different types of antibodies can be produced by the marine organisms. It's antiviral, it's anti-cancer, and against the human pathogenic bacteria. And second one is the hydrocarbon dehydrations. There is possible link between the nitrogen cycling processes and the hydrocarbon dehydration in the marine systems. So it is also important. And I am coming to the closer to my lecture. These are some marine research institute present in the India. It's a Central Marine Fisheries Research Institute, Kochi. CSI, National Institute of Oceanography, Goa, and others two, one from the Kolkata and another Bombay. There is another research institute of the Bhavnagar, Gujarat. And what are the conclusion of this lecture? It is that the 3/4 of our planet is covered by the water, and 97% of the total Earth is marine, that is covered by the water, out of total water. And the marine microbes is to some difficult for the studies from the other systems, and marine microbes are extremely reasonable, extremely difficult to study, and which should be explored. Because they are the resourcefuls. They are the resourcefuls area. They are the resourcefuls area. Most of the secondary metabolites are produced in these stress conditions. So, if we successful in tapping this resourcefuls microorganism, it will be better for the human mankind. So I acknowledge Mrs. Chitita Chatterjee, who is my research scholar of the Presidency University. Actually, she prepared the total PowerPoint presentation. That's why she must be acknowledged. And thank you all for the present hearing discussion. Thank you all.
Thank you, sir, for your deliberation. Your lecture is started from the evolution of life on Earth, and it is in a very lucid and easy way. Life starts from water, and you explain all the sea zone where life exists. The vastness of ocean is always tops the list of human inquisitiveness. You mention the bacteria, fungi, algae, virus, protozoa, and all the sea microbes. You also mention halopelagic zone, and that is unknown zone, and deep sea zone also, which is 70% of all ocean water, but why it is so strenuous to all the living beings. You also mentioned, sir, all the inhabitants of hydrothermal vent region and largest and smallest microbes within the sea water. You also cover the basics about the bacteria, the marine fungi, marine algae, and many more. And this presentation is really informative for us. I think all the participants enjoy the lecture, and the chat box shows its reflection. You also mentioned, sir, that aerobic bacteria or facultative anaerobic bacteria are the main inhabitants of salty sea water. And also you mentioned that actinomycetes are inhabitants of marine water, and there is a prospect of research. And and you also mentioned that virus are really important creatures of seawater, and your lecture is really informative, sir. And not only it is for biology students, but anyone who is interested on science. Actually, for all of us, we enjoyed the session. Today, really, you describe all the vastness of marine microbial biodiversity. I am really thankful to you, sir. And now this is question answer session. If anyone have question, please write it in chat box. Any question, participants, you can write in chat box. No question. No question, and only they thank you. They bid thanks, and right here, excellent presentation, wonderful session, but no question is there, sir.
Thank you, madam.
I really enjoyed the total session. Very good presentation, and within a very short period, you mentioned all the microorganisms of seawater, and their importance and research area, and lack in research, why marine microbes are in problem for doing research or ongoing research, and research institute also. You mentioned all the aspects, sir. Really, we enjoyed the session, sir. Thank you, sir.
Thank you, madam. May I leave?
Sir, one announcement is here.
Yes, sir, you can leave, but if no problem, you can stay.
Sir, one announcement is here. We are going to organize one conference on biodiversity in the month of March 2021 from 26 to 28. So we will discuss about the conference. Only 5 minutes is enough to just discuss and to show the PPT slides. Can I proceed, ma'am?
Okay.
Ma'am, can I proceed?
Yes, you can proceed. No question in chat box, so you can proceed.
Hello.
Hello. National conference on visible?
Yes, Yes, sir, visible. Please proceed, sir.
Okay.
Yes, it is visible.
National conference on biodiversity conservation
online platform, 3 days conference, that is 26 to 28 March 2020. Topic: General account on biodiversity with special reference to status and conservation strategy. This is the topic. This is the main topic. Organizer same Flora and Fauna Asia Facebook group, Department of Botany, Seva Bharati Mahavidyalaya, Kapgadi, Jharkhand, West Bengal, and Drishtikon Organization, Panchkura, Purba Medinipur, West Bengal. Important dates, that is dates of conference 26 to 28, that is 3 days conference, online mode. Last date of registration is 31st January 2021, time 10 p.m. Subdisciplines, that is animals, plants and microbes. From animals, chordata and non-chordata, plants, flowering and non-flowering plants, microbes, bacteria, cyanobacteria and fungi. That is limited here. Category of participants, category A, that is faculty, scientists, scientific officers, teachers and
state-aided college teacher, and so on. Category B, that is research scholars, research assistant, scientific assistant, and staff member. Category C, postgraduate students of any discipline with sound knowledge on biodiversity conservation. Category D, that is UG students, with others, school students having strong knowledge on conservation strategy. Presentation and mode of presentation, that is online, Google, Zoom, Cisco WebEx, Jitsi Meet, preferably that is Google Meet and Cisco WebEx. Types of presentation, oral and poster, selection as per the recommendations of the committee. Important announcement here, that is participants should register their names through the link provided by us and submit their abstract within 150 words. All will join in a group, that is WhatsApp group, like that our biodiversity webinar, like 1, 2, 3, 4 and so on, which is mandatory. We will provide all the information separately through WhatsApp group. After registration, all will get message separately. Full length paper should be submitted within last week of February 2021. No registration fees, but during publications of article, a nominal publication charge is applicable. We will publish article either bookish form with ISBN number and journal form with ISSN number. After peer review process, publication charge will be applicable if you wish to publish your article. Guideline for abstract. Abstract should be in New Times New Roman 12 PPT with 1.5 line space. There should be title, author's name, designations and affiliation, email ID, text and keywords relevant to the paper. Full length paper or review article or short notes should be placed to the committee before 30th January 2021 with figures and tables if required, but not more than 4 A4 size pages. Registration link is here. Please go to the group. Contact number if required, that is available in the chat box and even in the WhatsApp group and Telegram group. So please go to the registration for conference on biodiversity in the month of March 2021. Thank you.
Dr. Debobruta Das, I am asking you or requesting you to deliver vote of thanks.
Ma'am, Raj sir is there.
Raj Shankar Das, sir, are you present here?
Yes, ma'am, I am present.
Yes,
I'm
then
present.
vote of thanks. Please deliver vote of thanks.
Yes, sir, start.
Sir, thank you very much for delivering such a wonderful, for giving us such a wonderful session on marine microbial diversity and its importance. Thank you, Dr. Debobruta Das, admin Flora and Fauna Asia Facebook group, for organizing such a wonderful webinar series named Biodiversity, and it is webinar number 16, and we, all the participants, are getting opportunity to attend these webinars, aware about such a wonderful sessions and know about the biodiversity and the different aspects of biodiversity. Thank you, Dr. Ampi Ghosh ma'am, for organizing such a wonderful session and giving me the opportunity to deliver, to introduce today's resource person. Thank you, Dr. Rabindranath Bhattacharjee sir, for giving such an enlightening and illuminating session on marine microbial diversity and its importance.
Thank you, Professor.
Oh,
And
thank you,
hopefully
Rajshankar Das sir.
Hopefully we will wait for such webinars in future as well. And Dr. Devabrata Das sir has announced for the forthcoming conference. And we are really happy that we will be able to attend the conference as well. And I am participating in this webinar series from the first day webinar, Biodiversity Webinar 1. And I can see it's particular in every aspect, starting from registration to giving us all the notifications regarding the webinar and giving us a reminder about all the important events. Thank you very much. And thanks to all the participants, above all, for attending today's session. I hope we all gathered some important information, know some important information about today's webinar. Thank you very much. Thank you.
Thank you, sir.
Thank you, Rajshankar Das sir. Thank you for attending all these webinars, and you are such an enthusiastic participant, and really I am thankful to you, and all the organizing members also enjoy your talks. Thank you. Sir, now you can leave.
Okay. Thank you, madam.
Yes, sir.
Thank you, thank you.
Thank you, sir. Good night, sir.
Good night.
Dear participants, if you have any explanation or any questions or any query or about any kind of that is I think explanation or any kind of information, you can say something about before the audience. Dear valued participants, Sauji sir, Bijoyji sir.
Hello. Hello, Dr. Das.
Yes,
Hello,
yes.
good evening.
Yes. Sir, say something about.
Yeah. So, yeah, it's a very nice session. I have enjoyed the entire session. So, kindly share those for the 3 days workshop. Kindly share it so that I can pass on to all my fellow colleagues here in Bhubaneswar, so that they can submit abstract and papers for the 3 days workshop. Kindly share it in my email.
Yes, sir. And dear participants, please convey the message to the others that in this way we can organize a great that is conference on biodiversity on various aspects.
Yes.
Obviously, sir, so we will post the link and details in the group WhatsApp and that is Telegram group, and even we will post that is in different that is I think e-services, and I will obviously give you the link and others to your mail, sir.
Okay. Thank you.
Thank you, sir.
Huh?
Anil Kumar sir, do you want to say something? Anil Kumar sir? Anil Kumar sir? Can you hear me? Sir, do you want to say something?
Yes, ma'am.
Yes, sir. Please
Yes,
proceed.
madam. I want to know
what are the specific adaptation of biodiversity of bacteria
Sir, resource person left the room. If you have any question, please put it in your feedback.
Madam, I have already written the question in a feedback form.
Okay, sir.
Yes.
Okay, sir. We will convey.
Okay, thank you, madam. Thank you.
Anyone? Participant? Dr. N. Pankaj. Dr. N. Pankaj.
Yes, sir. I want to know about your future conference
Yes, sir.
regarding. I am working on amphibian diversity of Bihar.
What kind?
So, is there any scope for this topic in your workshop?
Say your topic, your field research.
My field of study is amphibian diversity, sir.
Oh yes, no problem. No problem.
May I submit my research paper also?
Obviously, sir.
Why not?
Okay.
Actually,
Okay,
we are going to publish in international journal.
Share all the information on a group.
It will be in a group, sir. Please wait for today. After finishing this webinar, we will give the link in the group. Please wait few minutes, sir.
Okay, ma'am. Thanks for organizing such a webinar, ma'am.
Please stay with us. Thank you, sir. Thank you. You are here. That's why we are here.
Now we are giving feedback, and today's webinar feedback will be active. That is, the link will be active up to 9 p.m. today. So just give your valuable feedback. তুমি লাইনে থেকো, বুঝলে তুমি লাইনে থাকো, আমি তাহলে বেরিয়ে যাচ্ছি এখান থেকে। লিঙ্কটা নিয়ে নাও, যদি লিঙ্ক দিতে হবে দিবে।